start. Everyone, quiet please. Quiet please. Quiet please. So good afternoon everyone and welcome to my course Genetics. Today is a rainy afternoon and instead of stay at home, rolling in our bed, enjoy the beautiful moment, we have to come here and torture each other. So at least make this time valuable for you. So in Thursday, we have discussed with each other on the principle of mitosis, on how the cell go on through the cell division with all the duplication, the DNA, and all the checking with the cell cycle checkpoint. So mitosis, in short, is cell division that keep the same genomes, and as a consequence from one mother cell we produce two daughter cells with the genome exactly like the mother generation. Mitosis is happening mostly in somatic cell and also in the gametophyte cell at the early stage. Okay? During the cell cycle, mitosis in the mitotic phase composed of four main parts. First is with phase, prophase, metaphase, anaphase, and telophase. The interphase is not counted as one phase mitotic phase. Okay? And during the cell cycle, there are many different checkpoints, but there are three main checkpoints, which is first is the G1 as checkpoint. Okay? Second is the G2M checkpoint, and the last one is the M checkpoint, which is also called as the spindle checkpoint. Let's watch a small brief overview on the mitosis and the cell cycle.
An adult human contains an estimated 100 trillion cells, and yet we start life as a single cell. To grow, develop, and repair tissue damage, we rely on cell division. In eukaryotic cells, this process is accomplished by a series of well-orchestrated steps called mitosis. Every day our bodies must produce millions of skin cells to replace those lost through normal activity. Each of these cells must have a complete complement of the genetic material. Prior to cell division, where the cell divides into two identical cells, the DNA needs to be replicated so that each daughter cell receives an exact copy. Following DNA replication, the chromosomes condense in the nucleus of the cell. DNA condenses by wrapping around cores of histone proteins, forming nucleosomes. This beads-on-a-string structure is called chromatin. As a cell prepares to divide, chromatin coils up further, shortening and condensing the chromosome. The replicated chromosomes are called sister chromatids. The replication of DNA and the formation of sister chromatids is one part of the entire cell cycle. To prepare for cell division, the cell goes through interphase, which can be divided into three distinct phases. During G1 or GAP1 phase, all the organelles and cytoplasmic components, including the centrioles in animal cells, replicate. Then during S or synthesis phase, the DNA replicates. Finally, during G2 or GAP2 phase, all the enzymes needed to aid in the process of cell division are produced. Most eukaryotic cells spend a great deal of time in interphase, and a very short period of time actually dividing, a process called mitosis. The cell is now ready to go through mitosis, which consists of prophase, metaphase, anaphase, and telophase. During prophase, the chromosomes condense and become visible, appearing as two sister chromatids held together at the centromere. The cytoskeleton disassembles as the spindle begins to form. In animal cells, centrioles play an important role in the distribution of the chromosomes in the dividing cell. The centrioles migrate to opposite poles, establishing a bridge of microtubules called the spindle apparatus, and the nuclear envelope breaks down. Towards the end of prophase, chromosomes attach by proteins in their centromeres called kinetochores to microtubules from each pole, moving the chromosomes toward the equator of the cell. During metaphase, all chromosomes are aligned at the equator of the cell, called the metaphase plate. Anaphase begins with the degradation of proteins that hold sister chromatids together, freeing individual chromosomes. The free chromosomes are then pulled by their kinetochores to opposite poles. At telophase, a cleavage furrow forms in the center of the cell. This indentation is made from a constricting belt of actin filaments surrounding the inside of the cell's circumference. Chromosomes cluster at opposite poles and begin decondensing as the nuclear envelope reforms around them. The spindle apparatus disassembles as the microtubules are broken down into tubulin monomers that can be used to form the cytoskeleton of the daughter cells. In animal cells, cytokinesis completes cell division by extending the cleavage furrow to completely separate the newly formed daughter cells. Since plant cell walls cannot be constricted by actin fibers, vesicles form an expanding membrane partition called the cell plate. Like animal cells, plant cells use cytokinesis to finish the division of the contents of the cytoplasm between the two identical daughter cells. During the cell cycle, certain checkpoints are encountered to make sure the process is occurring accurately. And if it is not, the cell cycle will stop at the checkpoint and correct, or possibly inhibit that cell from dividing. The first checkpoint is the G1S checkpoint, and is considered the primary point at which the cell cycle continues or stops. External signals and growth factors can influence the cell cycle and affect the progress at or before this critical checkpoint. The G2M checkpoint allows cells that have successfully completed all three phases of interphase to begin mitosis. The last checkpoint is the spindle checkpoint, ensuring that all chromosomes have attached to the spindle in preparation for anaphase. Growth factors, the size of the cell, and the nutritional state of the cell are all contributing factors in cell cycle regulation, ensuring that only certain cells divide at appropriate times. Once all the checkpoints in interphase are cleared, mitosis can occur. From interphase to cytokinesis, the entire process of cell division can take on average 10 to 20 hours in a typical plant or animal cell. Depending on the nature and use of the cell, the process can happen at different frequencies as well. In humans, our skin cells have a high turnover rate due to wear and tear, and go through mitosis very frequently, whereas other cells, such as adult neurons and muscle cells, rarely divide. The accuracy of mitosis, as well as the consistency of the checkpoints during interphase, ensure that most cells in a eukaryotic organism can produce identical copies of themselves. This process allows for growth and repair to prolong overall physiology as well as life itself.
forgot to talk about the DNA replication. So, just a quick slide about the DNA replication. So DNA replication, first of all, is always happening from which direction? The newly DNA strand was only synthesized in which direction? 5' to 3'. Okay, so in DNA replication, when the cell have a signal for the DNA to replicate and turn it to the S phase, so about the G1 and S checkpoint, that if the cyclin was crossed, the checkpoint is returned to the S phase where DNA was synthesized. So when the cell was signaled for DNA to synthesize, first of all, the helicase would come and break down the hydrogen bond link between the two single-strand DNA together. Next, we will have the complex protein will keep the two single-strand DNA not binding together again. And then what we have a kinase, which are the RNA primer into each DNA strand. Where the two strands, one is a leading strand, and the other strand is called the lagging strand. Because in the leading strand, which is have the direction from 5' to 3', the newly DNA strand will synthesize continuously. And the lagging strand, which have the direction from 3' to 5', the newly DNA strand will synthesize discontinuously into discontinuous fragment that we call the Okazaki fragment. Okay? So afterward, after the RNA primer, we added the DNA polymerase 3 will, DNA polymerase will add the new nucleotide to primase to form the DNA strand, the newly DNA strand. Okay? Afterward, the exonuclease will erase the RNA primer and replace by the DNA strand. And finally, we have ligase which will seal up the Okazaki fragment to form a newly DNA strand. Okay? So that's the whole process of DNA replication we need to know. And of course, when DNA replication, it will provide mutation. And we will have a chapter talking about how mutation will form and how the mutation was spread out in the population to form variation or polymorphisms in chapter 4. So now, just a question to reboot your brain. The G1 phase of cell involve which step of the following step? 1. Prepare DNA for replication. 2. Replicate the DNA. And 3. Prepare for the mitosis phase. Yeah, please.
Okay, so the G1 phase prepares DNA for replication. Meanwhile, the S phase is where DNA replication takes place, and the G2 phase is for mitosis preparation.
That's correct. My solution is just a little bit different. So the whole interface is prepared for mitosis, but your answer is also true. The G2 is also prepared for mitosis. And then from this mitosis, this is a figure about mitosis process. So could you put the genetic term in the blank for me from 1 to 9? So 1, 2, 3 is detailed in the picture, and 4, 5, 6, 7, 8, 9 is a different thing. Yeah, please.
One is interface and two profiles. One is is the interface and is profile and three is metaphase and four is anaphase.
Do we have another answer, please?
Can I use the laser? Okay. So first, the one is this black is a chromosome. Inside here we have the 2 is the chromatin. So this square is the interface.
But the one is outside of the
Mitochondria.
this is produced.
Okay, so the one is the central. The 2 is chromatin. Okay, so in here is interface. Interface here. This prophase. Here is metaphase. This is metaphase, and for this tetra. This 4 stick. Okay, so next up is
Three is microtubule.
this is microtubule. Yeah. Okay. No, hold on. Không sao đó là cái này. Đây gì? Cái này điền rồi đó là cái này theo là cái bây giờ cái
7 is
yeah 7 is cái này anaphase.
anaphase.
Yeah. The 8 is the telophase, and last one is 2 single cell. Thank you for helping.
Thank you very much. Correct. So we have different phase, mitotic phase, prophase, metaphase, anaphase, and telophase. And cytophase, one cell divide, we also call it cytophase. Okay. Okay, so there are also the question on what is different in the chromatin and chromosomes. Okay, so chromatin is mentioned about the complex between DNA and protein histone, and the chromosome is when the protein start to pack up to twist again to form a compact, and then form of the chromatin is what we call chromosome. Now come to next process is meiosis. So meiosis is the form of produce of gamete cell, which is happen in gamete producing cell, mature gamete producing cell. So during meiosis, it composed the same with mitosis. It's composed of one time the DNA will replicate. The difference with mitosis is involved 2 times the cell divide. So we will have meiosis divide phase 1 and meiosis divide phase 2. So as a consequence, after meiosis will finish, we will have 4 daughter cells produced with each daughter cell will have the number of chromosomes was decreased by a half compared to the parent. And due to the cross-link, this chromosome is not really identical with their parent. So this is a figure of pipeline where the meiosis was occurring. So from one parent cell, which is a cell with 4 chromosomes here, 2 paired chromosomes. After prophase 1, we will have the same with meiosis, but different with mitosis is in metaphase 1, we will have quiet place, please do not noise. Instead of one line of double chromosome, rather is the metaplate, we will have 2 line chromosomes who was gathered in the metaplate, which is the equator of the cell. So after anaphase and in telophase 1, we will have 2 cells with the number of chromosomes will decrease by a half, but this is including the number of chromosomes. And during the metaphase 1, we have also a phenomenon called crosslink. Okay? On crosslink, we will study the mechanism of softening more in chapter 6 related to the Morgan rule of inheritance. For that, we know that in the crosslink, we will have part of chromosomes, two homologous chromosomes will exchange the region to each other. So in here, for example, we have a part of the red chromosome here will exchange with the blue one. So as a result, here we have part chromosome is recombinant product compared to the mother cell. The meiosis 2 will happen just like mitosis. In the prophase, the membrane disappears. In the beta phase, it gathers 1 line in the equator of the cell. And in the anaphase 2, in telophase 2, we will form 4 of the cells with the number of chromosomes was decreased by a half compared to the cell. Okay.
Have you ever wondered why children do not look exactly like their parents, or why some siblings look so different from each other? The answer lies in the process of meiosis, which generates the gametes for reproduction. Meiosis is the process by which haploid cells are produced from a diploid cell. For this to occur, the chromosomes must be correctly sorted and distributed in a manner to create genetically unique cells with half the number of chromosomes as the original cell. Meiosis occurs in special cells called germ cells within the gonads of males and females. For the successful reduction in the number of chromosomes to occur in the new haploid daughter cells, 2 rounds of divisions are necessary. These divisions are termed meiosis 1 and meiosis 2. Although the processes of mitosis and meiosis look similar, there are a few major differences. One obvious difference is that mitosis results in 2 diploid daughter cells, and meiosis results in 4 haploid cells. As in mitosis, meiosis begins after a cell has successfully completed the G1, S, and G2 stages of interphase. During the S phase of interphase, the DNA is replicated, producing 2 copies of each chromosome called sister chromatids. The paired centrioles in the cytoplasm duplicate and begin extending microtubules that will form the meiotic spindle. Sister chromatids remain attached at the centromere and condense as the cell enters prophase 1 of meiosis. Up to this point, the cell looks similar to mitosis, but 2 events occur in meiosis that do not occur in mitosis and lead to genetic diversity. The first event occurs during prophase 1, when homologous pairs of sister chromatids lie side by side in a process called synapsis, forming a tetrad or bivalent. The homologous chromosomes share similar, but not necessarily identical genes. Once this structure is formed, the second event, called crossing over, can occur. During crossing over, a physical exchange between chromosome segments of non-sister chromatids occurs, increasing genetic diversity. Prophase 1 concludes with the fragmentation of the nuclear envelope as the duplicated centriole pairs move to opposite poles of the cell. As they move, the centrioles extend spindle fibers, forming the meiotic spindle. In prometaphase 1, with the paired centrioles in place, the meiotic spindle is fully formed. The sister chromatids attach to the spindle fibers by their kinetochores. Here, another key difference between mitosis and meiosis occurs. Because of synapsis and crossing over in meiosis, homologous chromosomes remain aligned, so that a pair of sister chromatids is attached to only one pole by the kinetochore microtubules. In mitosis, each sister chromatid is attached to a spindle fiber. The fibers originate from opposite poles. During metaphase 1, bivalents randomly align along the metaphase plate. Due to independent assortment, this alignment is random and adds to genetic diversity. In anaphase 1, the homologous chromosomes separate and move toward opposite poles. Meiosis 1 ends with telophase 1, when the chromosomes decondense and the nuclear envelope reforms. Cytokinesis separates the cytoplasmic material, and the 2 daughter cells are separated by a cleavage furrow. Since the final product of meiosis 1 is 2 haploid cells, meiosis 2 begins without the chromosomes going through another round of DNA replication. Centrioles again duplicate and begin moving to opposite poles of each cell. In prophase 2, the sister chromatids condense while the spindle starts to form as the nuclear envelope disappears. In prometaphase 2, the sister chromatids attach to the spindle by kinetochore microtubules, with sister chromatids attached to opposite poles. The spindle aligns the sister chromatids along the metaphase plate during metaphase 2. During anaphase 2, sister chromatids separate and individual chromosomes move toward the poles. The entire process ends with telophase 2, as the chromosomes decondense and the nuclear envelope reforms. Cytokinesis occurs and cleavage furrows separate the 2 daughter cells into 4 haploid daughter cells. The haploid daughter cells will specialize into gametes, either sperm or egg. These fuse in fertilization to form a zygote, which will grow into a child. The child receives half its chromosomes from its mother and half from its father. Because men and women produce millions of gametes, and the selection of gametes in fertilization is random, this contributes to genetic diversity. This explains why a child isn't identical to either parent. But why aren't siblings identical? The diversity comes from several sources. In prophase 1, non-sister chromatids can exchange DNA through crossing over, increasing the genetic diversity of individual chromatids. In addition to crossing over, in metaphase 1, the pairs of homologous chromosomes align independently along the metaphase plate and sort independently into the daughter cells. This process, called independent assortment, produces 4 genetically distinct haploid gametes. As the total number of chromosomes increases in an organism, the number of genetically distinct gametes increases by 2 to the nth power. This means an organism with n equals 3 can produce 8 unique gametes. For humans where n equals 23, there are 2 to the 23rd power unique gametes formed, or over 1 million different possible combinations. The combination of independent assortment, crossing over, and the random pairing of gametes during sexual reproduction increases genetic diversity and explains why a child will not look exactly like his or her parents or siblings.
Okay, so one thing interesting about meiosis is the distribution of chromosome in each gamete is totally random. Okay, we don't know among the 2 chromosomes in the pair, we don't know in each gamete which chromosome will be distributed in. So as a result, we will have a combination. A number of combinations will increase depend on the number of chromosome pair. For example, in Drosophila, we have the number of pair chromosome is 4, which is 4 pair of chromosome in their genome. So in meiosis, we will have number of combination as 2 power by 4, which is 16 chains of combination in the gamete. And we have not counted into the cross-link yet. So if we have a cross-link, there we will have a number more chains randomly when we have different kind of combinations. So the variation in the genetic background is also a fundamental source for evolution, right? So just compare between mitosis and meiosis. So in mitosis, we have here a daughter cell will have the same genetic background with the parent cell. But in meiosis, we will have daughter cell, 4 daughter cells, but with the genetically, first number of chromosomes was decreased by a half. It's a half-lived cell, not a deep-lived cell. And secondly, we will have, due to the genetic cross-link, we will have this combination will be different also with the parent cell. And this is different between the male gamete and the process is a little bit different between the male gamete and the female gamete. So in the male gamete, it happens like regular meiosis, when from one gamete producing cell, we produce 4 sperm cells for male gamete cell. However, in the female gamete, which is the ovule here, due to the amount of cytoplasm are needed to mature female gamete here is quite large. So during meiosis, we will have the oocyte, all the cytoplasm, including also the mitochondria and all the other stuff, with largely span for 1 cell here, and during meiosis 1 is largely. So this one will develop into a mature female gamete cell. Okay, so that means in female in general, in one parent, we will have only one gamete cell was mature. So I will have make a table compare between the mitosis and meiosis here. So now I have a small question. What is a true statement about meiosis? So, with all this statement, which one was true about meiosis? First, meiosis involved two sequential cell divisions. And two, no DNA replication occurs between meiosis because the chromosome size is half. Three, the crossing over leading to variation in the genetic material. And four, during meiosis, paternal and maternal chromosomes are separated and up into different cells. Which one are correct? Quiet, please. So, we are looking at our example for final exam. Final exam will compose from 40 to 50 questions like this. And you will have to handle it in 1 hour. Okay? That means that just around 1 minute per one question.
I choose statement 1 and statement 3.
Okay. Statement 1 and statement 3 is right. Anyone other have a different answer? So, statement 1 and 3 is correct. So, meiosis involved in two sequential cell division. We have meiosis 1 and meiosis 2, right? No DNA replication occurred before meiosis. No. It involved one part, the DNA was replication, DNA copy, okay? And 2 times the cell will divide. 3, crossing over leading to variation in the genetic material, that's correct. Because the new set of DNA, even if recombinant was formed during crossing over. And 4, during meiosis, paternal and maternal chromosomes are separated up into different cells. This is 4 because in meiosis, paternal and maternal chromosomes are divided independently. Okay, so we don't know in one cell we will have for each pair chromosome we will have from a maternal or paternal. So this process is happening rapidly. So let's come to the next chapter. I'm talking about transcription and translation. Quiet, please. So transcription and translation is interesting process where information will transfer from DNA to RNA to protein. But it's a little bit complicated, in, especially in a cell, where we have not every gene will transcript and translate. In fact, in most of the cell, most of the genome was silent. That means they will not be transcribed and translated. Only a few parts of the gene was designed to transcribe and translated by the cell, which is necessary for the function of the specific cell.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of 3 bases in the nucleus. The DNA code determines the order in which the 3 bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
So, transcriptions, that's clear in terms of genetic. Transcriptions is the information will transfer from 2 types of acid nucleic, which is transferred from the language of DNA to the language of RNA. So this coding are more or less the same between DNA and RNA. So this process is quite simple, quite easy to understand. However, the next step, translation, is much more complicated. One is have to transfer language from the language of acid nucleic, which is RNA, to the language of protein, which is the sequence of amino acids. So we will have transfer from the 4-letter code to the 20-letter code. So just to remind you about the main difference between DNA and RNA. So in DNA we have the nucleotide is the sugar is deoxyribose sugar, and in RNA is the ribose sugar. While the carbon number 2, so we have carbon number 1, the alpha oxygen here, 1, 2, 3, 4, 5. And in the carbon number 2, instead of the hydroxyl, like in the ribose, we have a hydrogen atom. Okay? And in DNA, we have 4 nucleotides A, C, G, and T. T is stand for thymine. While in RNA, the 3 other nucleotides are the same, A, C, and G. Meanwhile, the uracil is just like the thymine, but it just replaced one methyl group by one hydrogen atom. So in the clip, they have produced typical transcript and translation in the eukaryotic cell. However, the process in the prokaryotic cell is a little bit different, because in prokaryotic cell, we only have the nucleus region. Okay? We didn't have the nucleus membrane to separate between the nucleus region and the cytoplasm. So as a result, after a transcription factor, the RNA polymerase starts to synthesize the mRNA. While the mRNA are being synthesized, the ribosome can come and binding immediately to the mRNA and start to synthesize the polypeptide. So we can have in the prokaryotic cell, the transcription and translation happen continuously in the same time, and it both occur in the cytoplasm. Okay. One more thing, in prokaryotic cell, the ribosome complex, which is combined by two subunits, the small one and the large one. The combination of the ribosome is 70S, which is contain the small one, small subunit is 30S, and the large subunit is 50S. Okay. Meanwhile, in the eukaryotic cell, we will have the transcription and translation happen discontinuously. So in the eukaryotic cell, the main difference is the nucleus membrane here. It separates between the nucleus region and the cytoplasm. So in here, after the transcription, the RNA was synthesized, we will have a process happen only in the eukaryotic cell, which is the mature process of mRNA. We'll talk about it later. After the RNA is mature, that means it has been capping the 5' cap, adding the 3' poly A tail, removing all the introns. It will go out to the hole in the membrane, the nucleus membrane here, and in the cytoplasm, it will catch with the ribosome complex to perform the translation. Okay, so in here, in eukaryotic cells, transcription is happened in the nucleus region, and the translation was happened in the cytoplasm. Okay, if not a continuous process like in the prokaryotic cell. And the other differences is the ribosome in here is 80S combined by two subunits. One small is 40S, and the large subunit is about 50S. So first, let's talking about transcription. This transcription process happening, we want to have a complex of enzyme called a transcription factor. So the transcription factor will recognize the original protein it needs to synthesize through a sequence called a promoter. So promoter is different promoter is differences process between different organisms. So, for example, in prokaryotic cell, in E. coli, we will have a sequence located before the transcription starting site, TSS, which is position +1. SS is transcription starting site. So this is where transcription starts. So in the prokaryotic cell, we will have two regions. One region is before the +1 positions here, about 10 nucleotides, which is combined by the sequence TATAT. And another reason is before this position, 35 nucleotide is the single bond, is PTGH here. So there's many kinds of promoter in the prokaryotic cell. For example, in coli we have D5, D7, the Kozak promoter, and many other type promoters. Each promoter will have special signals to the transcription level that they would have. Okay? So the promoter here is contain a special sequence so that the transcription factor can go and recognize this is the region it has to combine and start to synthesize the transcription level. So, how do we know what sequence is the promoter of the gene? So this is a complicated question. How does the scientist know which sequence is represent for a binding site of a transcription factor? So for example, in the human genome here, then we incubate it with one transcription factor. Okay? It will bind in different positions of the genes. And then they separate it. Start to share the protein into pieces, and then sequence the region that protein is binding in. So for example, we will have in first sequence the TTA, TC, GTA, C. In the second sequence here we will have TGA, CTA, TC. In the third sequence, we will have CGA, TC, TTC. Fourth one, we will have G, T, A, C, C, G, C, A, C. And the fifth one, which is So with the result like this, please identify for me which nucleotide is the most important nucleotide to recognize a promoter. A, this position, okay? C, and C, right? So we see that in 100% of the sequence, we will have the position of this one is A. This sequence is C, and this one was C. So that means that if one of these three positions change, the transcription factor will not bind in the promoter. Okay, so this three nucleotide could be a key key position for transcription factor. However, when I looking up this position, for example, I see that it only have two chains, T or G, right? Different division, one it could be both T, G, or A, but in this position only T or G. So that means if this position it changes to C, the promoter is divided by 2, right? Or this position, either it is G or T, or T or C. So with these positions, is also a key player to recognize the promoter region of transcription factor. So based on this, scientists can make algorithm to build up a binding motif for each type of transcription factor. And please remember that there are thousands of transcription factors. Each transcription factor will have a strand. Its own strand, its own specificity with different genes. Okay? So in a eukaryotic cell, so we will have of course, in the organization of the gene, we have the exon and intron here. So the TSS, the truncal side, plus 1 is here. And what typical eukaryote promoter will be a little bit longer than the promoter in the prokaryotic cell. So for example, we have TATA box, which is found in many promoters, okay? Which is have the position 25 nucleotide away from the transcription side, or CAAT regions, where the position is around 18 nucleotide before the transcription side. One sequence frequent to present in the promoter region of many genes is the GC island. So GC island is a term referred to a region that contains rig of G genes compared to the other. So it contains GC, GC, all along. So this GC island must play an important role in the epigenetic to control which gene was turned on and which gene was turned off during different situations. We come to this, return to this region in chapter 7. So, the transcription starts when the transcription factor starts to recognize the promoter and start to, when it's recognized, the promoter is binding and start to unwind the DNA strand to cut down, like DNA replication. It will break down the hydrogen bond and separate one DNA strand off. And different with DNA replication, where following the semi-conservative rule, we have both strands we're using as a template to synthesize a new DNA strand. In transcription, we have only one strand was using as a template to synthesize the mRNA. Okay? So, in here, the RNA polymerase bind to the DNA and start to synthesize the mRNA, come along during the length of the gene. And when it's finished, it reaches the termination regions, which is signal the transcription factor to release the mRNA, and the transcription process was finished. So let's watch the small clip. How transcription factor was binding into the
For RNA polymerase to successfully bind to a eukaryotic promoter and initiate transcription, a set of proteins called transcription factors must first assemble on the promoter. The assembly process begins upstream from the transcription start site, where proteins called basal factors bind to a short sequence in the promoter. Other basal factor proteins then bind, eventually forming a full transcription factor complex, able to capture the RNA polymerase. Basal factors are essential for transcription, but cannot by themselves increase or decrease its rate. A second set of transcription factors called coactivators link the basal factors with a third set of transcription factor proteins called activators. The activator proteins are not shown.
So, just remember that in transcription factor, in transcription, the transcription factor are not using both two DNA strands as template. This is only one template as a template to synthesize the new RNA strand. And because both DNA and RNA were synthesized from the 5' to 2'. So we will have in here, for example, in here I have a double strand DNA. So the transcription factor when binding in the top or in the bottom strand. In the bottom strand, correct. So it will binding in here and start to synthesize. That is what we call, why we call this DNA, this strand is a template strand, because the transcription factor will use this strand as a template to synthesize the new mRNA strand. So it starts to synthesize following the Chargaff rule. The complementary rule, I is linked with the U, stand for uracil, G is linked with C, and T is linked with A again. So the newly mRNA will have the sequence the same with the upper strand here, the top strand here. Okay? Only replace the T by the U. Okay? And that's why the top strand is called the DNA coding strand, because it has the sequence, the coding of this sequence is the same with the mRNA. And I have made a small compare between the DNA replication and RNA transcription here. So we see that the template in DNA replication is double strand, and as a consequence, we have the product is a double strand DNA. Okay, and in transcription we have a template is only one single strand. The transcription factor only using one DNA strand, and as a consequences, in the product we have a single strand RNA. Okay. The substrate in here is a deoxynucleotide 3-phosphate, and in here is MTP nucleotide 3-phosphate. Okay, to synthesize the RNA. The enzyme is DNA fold, RNA fold. And in DNA replication, we need an RNA primer. Meanwhile, in transcription, we did not need the RNA primer. Okay, so now we will have a small break, 5-minute break before we go further on. 5-minute break.
Ngày xưa tầm hồi mình đi học xong thì một lần mình trưa mình ngủ dậy mình cứ phát ngủ trắng luôn, rồi mình phải nằm xuống nằm xuống ngồi nằm đều khoảng tiếng rưỡi kéo đầu đấy cái đường cả phải lên phát kiểu quay kinh cái đồ quay nó chảy đi cái gì cả, xong rồi lại phải nằm xuống rồi cố đứng lên. Đối với đoán chứ gì? Đoán đi thì tất cả mình đoán chứ. Chữ Đức. Cho dùng 2 năm rồi. Trung ơi. Một nửa, nửa. Đây cho mày mượn. Được. Nếu tìm được cái thư FPT mà xem. Đây tao mở lấy máy tao. Làm đi. Tất cả. Cho tao vào cái chùm đánh bắt nào. Ở đây. Cái chùm có tay xin đâu. Nó đánh gôn. Để ra tao vào. Để hai thầu. Ra rồi cái chùm. Ra ngồi chỗ tao ngồi ấy. Đây. Tao ngồi ngoài đâu. Nước gì đấy? Nước gì đấy? Cái này nước rồi, con này nước bẩn của tay sau. Mới thì nó sẽ đấy. Ai bắt rồi đấy? Cà phê gói. Kiểu vậy. Cà phê tháng một nếu hết. Ăn đi sắp thì này lúc đầu mà đây có hơi đúng chắc hết. Thế à?
Không. Có bài tự làm. Mấy lời đâu anh em.
Đi mẹ. Con này có tên mà. Ngu ấy rồi. Vâng, biết điểm luôn ạ. Chưa biết điểm. Vừa nháo giờ mail về đã. Sáng
Chưa biết là nó ai không này.
Vì nếu mà có điểm cộng thì nó sẽ không được cộng vào chút. Còn được 50 và 35. Trong 7 rưỡi. Chắc lớn nữa. Nhưng mà nó thanh đến tới đó đủ. Nó thanh đến tới 17,5. 9,9 mấy. Đang tới 60 ta không phải lo về xe đường.
50 à? Trong slide thì nó có
Trong cái file ấy.
Okay, so some question has still wondering about the motive binding contractile factor. Well, so for example, in here I have a motive binding contractile factor. So like in here, normally the motif binding is around behind nucleotide. Okay? That's the technical reason, technical length for motif binding. So for example, in this motif binding, so we have this one is T, and C. That means that 100% of the case of the technical factor binding, we have the two nucleotides here is T and T. Okay? And the same situation with the C here and A here. So that means these four nucleotides is play a key role in binding transcription factor. Okay. However, we have a region like the position number 9 here. Let's say that 60% of this case, the nucleotide is A, and another, let's say 40% is a C. So that means that if the nucleotide here is also very important, but it can change between A and C. Okay, the binding transcription factor still binding in this region. However, if it's transferred to T or transferred to G, that this position, the transcription factor will not bind in it. So it has some matrix but it can be alteration between A and C, not like before here. Okay, before here, it changed the affinity between the DNA and the transfer factor was disappeared. Okay, and the same was in here. So in several positions, we will have different positions like T or C here. It can accept the nucleotide G or T in here, but not the others. Okay? So, in the phonetic cell, is the gene was organized as a specific structure called the operand. So operand is a combination of different genes, and in terms of genes, we have remind in chapter 1 we have gene is some fragment of DNA encoding for protein or maintain a specific function. Okay. In prokaryotic cell, most of the gene was encoding for protein. So in here we have different. For example, in here we have 1, 2, 3, 4, 5. 5 genes was organized as an operand. Okay, and it controlled by the same protein. So when it's activated, all the mRNA was synthesized, and we have a different protein with a similar function was composed.
Nuclear region here.
The green one is nuclear region where DNA was. So we have a promoter with 3 novels in here. And when the RNA strand binding in DNA is binding in the template strand, okay, we have the sequence complementary with the mRNA. And it will slide a little bit until it met the +1, the transcription starting site, and start synthesize the mRNA. And at the same time, with mRNA was synthesized, the ribosome will directly to binding with the mRNA and start synthesize the protein. So when the mRNA recognize the terminator regions, it will release the mRNA and finish the transcription factor. So how transcription factor was transcription was happening in eukaryote? So first, the promoter in eukaryotic cell is longer and in the further distance with the transcription starting site compared to the eukaryotic cell. And in here, so this is a process happen in the nucleus regions. So in the nucleus region, after the transcription factor transcription process, the pre-mature mRNA was synthesized. So with all the sequence after promoting, okay, on the starting site here. Then the pre-mature mRNA with following process. First is a 5' capping binding into the beginning of mRNA molecule. So this 5' capping have two effects. First, to protect the mRNA from a cleaving enzyme, and second, it a signal for the mRNA to get it out of the nucleus membrane through the hole in the nucleus membrane. Next, we will have a splicing which will remove the intron and also several extrons too. We will talk about it in the next couple of minutes why we have removed also the intron and extron due to the difference in splicing variant. And in the end, we will have enzyme to add the poly A tail here, and this poly A tail here also has an effect to prevent the cleavage of the mRNA. So after the whole process, the mRNA was considered mature and getting out of the cytoplasm through the hole in the nucleus membrane. So in here we have reasons. So in the genes of the cell, we have a promoter here. The transcription factor binding in here and start to synthesize from this position. This is a double strand DNA, that encoding point. So as a result, we will have the mRNA pre-mature mRNA was synthesized. So in this gene, because it's autotic cell, so we will have exon and intron, right? So this is the exon. And in middle is intron. I'm drawing not really accurate. Actually, the intron is much more longer than the exon. But for time being, that's where it's up. So the premature mRNA will contain also the exon regions and the intron regions. Lately, in the process, we will have the mature mRNA. First, it will have a capping. 5' cap. Okay. And it also have what is so-called a splicing process. When all the intron was removed and only the exon was here. And in the end, we have the poly A tail. And the whole process is happened inside the nucleus region.
Đây kỹ năng thì khả năng của cháu tiếp cận nhiều mắt.
After finish is go outside. Through a hole. And on the resting membrane. Get outside. And the ribosome start to synthesize the protein. So here is a DSS plus 1. And the amino acid is here from A to G. So here is a start codon, which is the starting site for translation. We have the polypeptide. So from this position, transfer starting side to this region where the start codon is the region what we call the 5' UTR, or stand for 5' untranslated regions. And again in here, when ribosome starts inside the polypeptide until it creates the start codon. For example, we have UAA here. Then this is the end of the polypeptide. So we will have a region here, not translated. We call it the 3' UTR. 3' untranslated. Okay? So for example in here, this is the transcription starting site. And this is the translation starting site. The distance between the two is 5' UTR. Okay? So for the student who participate in the bioinformatics practical class, you understand that the exons which coding in here is coding for mRNA, but it's not translated. So as possible, we will have the regions in the beginning, and in the end here is untranslated. Okay? It was transcribed, but it's not translated. So splicing can be different. As a result, one gene can have different transcription variation. So for example here, I have total from splicing side, we have exon 1, 2, 3, and 4. So typical splicing is an mRNA will be remove all the intron regions in here. Okay? So we will have this one including exon 1, exon 2, exon 3, and exon 4. But depending on the context, sometimes the cell will synthesize another variant, another mRNA variant. For example, in here, we have another variant which is the exon number 1 and exclude the both intron and also exon number 2. So in this RNA molecule, we have only exon 1, exon 3, and exon 4. For example, like in this case, we have a DNA, a gene, composed by 6 exons. We have the premature mRNA composed by exon number 1, 2, 3, 4, 5, and 6. However, when the splicing happens, it will compose, the classical one is because all exons 1, 2, 3, 4, 5, and 6, which here. Another combination is it compose exon 1, 2, overlap exon number 3, 4, 5, and 6. And the translation product will include only 5 parts. 1, 2, 4, 5, and 6. Another splicing variant, one, the exon number 2 and exon number 4 were excluded, and in the mRNA and the protein process, we only have the region number 1, 3, 5, 6. And last one, 5 please. We have here the exon number 2 was excluded. We have 5 regions, 1, 3, 4, 5, 6 in this order. Okay. So the alternative to splicing is really common in, especially in the very common in eukaryotic cell, especially in the higher organisms. For example, in one gene in Drosophila, the DSCAM1 gene, when you serve it in NCBL, you see that it has the chapter variant is more than 38,000 variants splicing. That means there are more than 38,000 splicing ways of this. So this is statistical between the number of genes and the corresponding number of RNA variants of those genes. So we have, in human, we have around 25,000 genes. We will have nearly 5,000 genes, only have one version, one variant of the RNA. So only one way the RNA will synthesize with all the exons. We have nearly 3,000 genes that have 2 variants of RNA transcription. And we have nearly more than 2,000 will have 3 variants, and so on. In here, for example, we have nearly 1,000 genes will have 10 variants and more. Okay. So this is an example of a transcription variant of the genes. So in here we have a coding gene FGFR2. So this gene is corresponding to the R syndrome, where the child, if they have a mutation on this gene, can have an intellectual disability, of course. The abnormal in skull development and leading to many limb, hand, toe, feet abnormal development. So this gene is composed by more than 15 exons, and we can see here it has many different transcription variants. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18 different variants, splicing variants. And among those, many regions was started in the first case. And lots are coding for protein.
As DNA is transcribed into RNA, it needs to be edited to remove non-coding regions or introns shown in green. This editing process is called splicing, which involves removing the introns, leaving only the yellow protein coding regions called exons. RNA splicing begins with assembly of helper proteins at the intron-exon borders. These splicing factors act as beacons to guide small nuclear riboproteins to form a splicing machine called the spliceosome. The animation is showing this happening in real time. The spliceosome then brings the exons on either side of the intron very close together, ready to be cut. One end of the intron is cut and folded back on itself to join and form a loop. The spliceosome then cuts the RNA to release the loop and join the two exons together. The edited RNA and intron are released and the spliceosome disassembles. This process is repeated for every intron in the RNA. Numerous spliceosomes, shown here in purple, assemble along the RNA. Each spliceosome removes one intron, releasing the loop before disassembling. In this example, 3 introns are removed from the RNA to leave the complete instructions for a protein.
So now the mechanism behind how RNA splicing can happen with the overcome of different exons, like in this case were not fully understood. But scientists have proved that the nearby nucleotide, for example, in this case, the nucleotide in the exon number 2, the barrier is the exon nearby exon number 2, and exon number 4, the intron here, may play a critical role for the activity of the enzyme that cutting through the intron and feed the exon 3. Alright, I think today's lesson is finished here. Do you have any questions? Okay, so thank you very much.